Yeah, so today will be about quantitation. So there will be another, this will give an overview today, and then there will be another lecture on quantitation uh, later that Kelly Ruggles will do, and that will be more on targeted quantitation. So basically if we look again at the experimental workflow, we have our sample and we lyse the cells, we might fractionate the proteins and then we digest them, and finally we separate the peptides with LC and then measure our mass spectra. And so what we do measure here is peptide intensities. And from that we want to deduce what the protein concentration is in our cells. So what we have here is that we have intensity of a peptide and we want to know the concentration of the protein in a certain sample I in this case. So during this workflow, every protein will go through uh, different losses. So if we know these specific losses and we know how well we can convert peptides into proteins, and then the peptide losses, and finally the conversion from a peptide to the signal. Then we can just do the calculation, and we could just simply multiply together the different losses, and from the protein concentration calculate the peptide concentration. And then in the case when we have different proteins that can produce the same peptide, we of course have to sum over those proteins. But I mean, we can't really measure all these losses, but what we can do is observe that if we keep all these conditions the same, then and we only look at peptides that can come from one single protein, we have a proportionality between the intensity of a peptide and the concentration of the protein. If we keep we have to keep the measurement within the linear range, which often, if we do the quantitation carefully, we do test for that it is within the linear range, but often when we do a global analysis, it's more assumed that that's the case. So one way that we can quantitate is to then first do a standard curve, so that we have a matrix that's similar to our matrix, our sample, but doesn't contain our peptide, and then we can spike it in and at different concentrations, so that we get we know how much we spike in, and then we measure a response. So we get this standard curve, which usually there is a linear range in the middle, and then at some point if we go down in concentration, we reach the limit of detection, and then at some point we get saturation at high intensity. So, but within there we have a linear range, and we can measure what if then subsequent samples, as long as we keep these conditions the same, we can then do measure from the intensity. We can go in and determine what's the concentration if we've done this. But of course, I mean, this we can only do for a very limited number of peptides. So and again, so what we assume here is that we have this proportionality. But if we want to measure a lot of peptides, what we can do is to compare samples. So we get a relative quantity for the same peptide in different samples. So what we then try to do is to keep all the conditions the same and repeatedly measure first one sample, then the second, and then third, and maybe in the end many. And what we get is, if we succeed to keep these conditions reasonably constant, then we get that the ratio between the intensities correspond to ratio between the concentrations. And this is what people call label-free quantitation. And it's used quite a lot. It works quite well if one can do the measurements over a short period of time. It gets more complicated if one measures hundreds of samples and it takes months to do the analysis. But even that people have done quite successfully. And the trick is then how to normalize properly between the samples that have measured. Because I mean, if you measure over months, the humidity in the air will change. It's difficult to keep the lab at the same temperature. You might have to change the chromatography column. So there's a lot of things that can change. So you definitely need to work out how to normalize well over all these samples. And we're going to talk more about, when we talk about data analysis, how to normalize. But just briefly, one assumption that one often can do is that the concentration of most proteins don't change in an experiment, and that's the ideal if one can have that assumption, so that there are just a few changing proteins. So what one can do is then, for example, take the median of the protein concentrations and say that that should be the same for all the samples. But there are more advanced methods to do that. But that it becomes very difficult to do this experiment if you have cases where most proteins change in concentration dramatically. Then one really has to think about how to do that properly. But that's actually not only for the label-free, but for any quantitative experiment, pretty much. So now the other extreme that we can do is to grow the cells in heavy and light medium. So the light medium, we use just the regular amino acids, but in the heavy we've replaced arginine and lysine with heavy arginine and lysine. So then in this case what we get is that since every peptide, especially if you use tryptic peptides and have labeled arginines and lysines, we can mix these samples at the beginning. So they only grow separately, but then we mix them, and then the assumption is that they behave the same throughout all the fractionation, digestion, and liquid chromatography and mass spectrometry. And we end up then with 2 isotope distributions for each peptide. And this assumption works well, especially if you use carbon-13 and nitrogen-14. That's definitely, people have shown that the assumption that they behave very well in the experiment, behave the same way in the experiment throughout all this, has been established. Now, the question is, how does these heavy isotopes affect the growth of the cells? And now, in most cases, for cell growth, it seems to work fine. There's no big phenotypical differences. But when you then try to make animals with heavy arginine and lysine, those are quite sick and have a lot of problems. And the main reason is that although chemically they look the same, because I mean all you change is the number of neutrons. The problem is that the change in mass will affect every enzymatic reaction just a little bit, and taken together it will, when you have a whole animal, it will lead to problems. but those are the assumptions, and they are not.
How much exposure they get sick?
So yeah, I mean, so what you try to do is to have 100%. I mean, that's not always. You have a distribution, and maybe some proteins are only 90% or 95%, but it's a lot, because otherwise it's very difficult to do the experiment. Yeah. The other thing one can do here is also have several different types of heavy. So you just replace different number of carbon 13 and nitrogen 15. And then you can do experiment with several experimental groups and mix them and then analyze them together. Oops. So the basic assumption is that really, if you succeed to do the mixing and if their growth is not affected by the heavy labels, then everything after that should be they experience the same losses. So here you should get very accurate quantitation, and much more accurate than in the label-free case, because you all these steps are.
If you do reciprocal, like instead of doing light with this, heavy with this, do you get approximately the same results?
Yes, but most people do that experiment. So you do both, preferably. So you both have your wild type and your control and your experiment, and label one first and then the other and compare. And usually you would only consider the proteins that give a reproducible ratio in those two cases. So that's definitely advisable to do, because there could always be some strange effects, and especially from contaminants. So if you get contaminants, they will be light. So that's why you can sort those out by doing the yeah, because they will not have consistent ratios when you switch. So this is just one example for where on the same experiment we looked at both with label-free and with metabolic labeling. And so this is just the same sample and then measured twice, either by metabolic labeling or by label-free quantitation. And you see that the distribution is much wider for the label-free, as we would expect, because there are many more steps that we need to be careful at keeping the same. And it's just not. But of course, this how big this difference is always depends on how well we can keep the conditions. So this is just one example of that. But what we see here is that we'll always get more variation in the label-free case, because there are more places where we can have variation. So now this just shows that if we do replication in the label-free case, we can get better. So what was done here is compared to, so this is the experiment was to do first IP on phosphotyrosine, and then fractionation and digestion, and either just one replicate of each or 3 replicates of the IP and 3 replicates of the fractionation. Then if we do more replicates, we see that the label-free becomes more accurate, because when we do replicates, we take out the variation. But we still have a much wider distribution than in the case of Silac.
for the top selected, the ones that are
most abundant.
Most proteins, the proteins that are selected at the top.
Yeah, that the most abundant. Yeah.
Detected proteins detected by two methods.
Yes.
They are similar.
Yes, they're similar. Yeah, they're. Yeah, they're pretty similar. I mean, it depends on. Yeah, if they're done in the same lab with the same instrumentation, they're usually quite similar. So then, what we're going to come back to a little bit later is what we then. How do we determine what's significant? And you'll see in many cases that people set some kind of arbitrary threshold on what's significant. Let's say they select all the proteins that change more than a twofold, but it's really not the right way to do it, because you should measure really what's your variation when you measure identical samples over and over again to see what the distribution looks like. And like in these two cases, if we arbitrarily set the threshold where at these dotted lines for what's significant, then you see in the two, the label-free and the metabolic labeling experiment, it means very different things. Because here we get, so what we should do is to integrate from here out on both sides to see what fraction ends up here at random when we measure identical samples. And that's going to be a very different number in these two cases. So the main thing is that the best thing is to see what your variation is in this specific experiment, and then use that to decide on the what cutoff to use, and also preferably in biological replicates, so that you get the because almost always when we do these experiments, the technical replicate reproducibility is much higher than what we get for biological replicates. So if we should really do this for biological replicates. In this case, we did this just shows for technical replicates, but still, that's definitely recommended. So I think we mentioned a little bit about protein complexes before. So one thing that we want to know about protein complexes, when we do affinity mass spectrometry, we usually label either have put a genomic tag on one protein in the complex, or we have a good antibody against it, and then this is in a complex mixture. So when we do have a pull down, what we end up with is both that we get some contaminant proteins that are sticking, and we also lose some parts of the complex. So so we both have incomplete information, and then we have extra. But one important thing is to figure out which ones are these non-specific interactors. And one way we can do this is by using heavy isotope labeling. So the way that people have shown how to do this is you have your tagged protein in light medium, and the wild type in heavy. And then you mix them together, and then do the immunoisolation of the mixture, and then do mass spectrometry. So what you expect is that for the specific interactors, they should only come from the light in this case, while the non-specific should come from roughly equal intensity from the heavy and the light. And then again, in this case, you should also always do the reverse also, to make sure. But what you, and that's actually what it looks like. So you get these specific ones, and then you get non-specific. Now the issue here is that when we have very fast exchanging proteins, so in complex that this it works really well for stable complexes, but you always have some parts of the complex might be fastly exchanging, so they will exchange in this time after you mix, before you do the immunoisolation. So, and then these will tend towards the non-specific. So they will, fast exchange will look like they would be non-specific. So you lose those. So, but what that means that is that this is a conservative method. So the ones that actually turn out to be specific, look like they're specific, they really are specific, but you lose some of them, the fast exchangers. Another thing that you can use heavy isotope labeling is to look at protein turnover. So, for example, you start with growing your cells in heavy medium, and then at some point you switch to light medium. And then you look at, as a function of time, how much light and heavy you have, which is what was there from the beginning, and then light is what's newly produced. So what you can do then is you can calculate the turnover. So these are the equations. So the assumption here is that everything is heavy in the beginning, and then it will, if you add up the heavy and light for each at each time point, you always have the same concentration. So this the assumption here is steady state, and then the change in the level of the heavy is what comes from the turnover. So you can, so it's should be in the steady state a simple exponential decay, and from there you can calculate the time that it the turnover time. So here TC here is the average time it takes for a cell to go through the cell cycle, and KT is the time it takes for half the proteins to turn over. So this is something that, and the nice thing with this is that it can also be applied globally to all the proteins in the cell. So now it's not always you can't always do heavy labeling of at cell growth. For example, if you have clinical samples. So then what people have, this is comes from Matthias Mann's group, this suggestion is to use CELTA. So for example, what they showed, if you want to look at breast tumors, you can take a bunch of breast tumor cell lines and then mix them together and use this, they call it super SILAC. So you take all these different cell lines and grow them in heavy medium and mix them into your breast tumor. And what you're then are hoping for, and which they've shown that it's actually the case, is that for a lot of the peptides that you see in the tumor, you have representatives in the mixture of cell lines. Yeah. So this is one way to do it. Another way to do it is if you're analyzing a lot of breast tumors, is to, let's say you have 100 breast tumors that you want to analyze, you take a little fraction of each of those and mix them all together, and then use that as a labeling.
Sorry, I didn't understand the Silac. I mean, this is the amino acid?
Yeah, so sorry. So Silac is the metabolic labeling.
Okay.
So where you add
I mean, what is the difference? Why does it work?
sorry, what is the difference between?
Like the heavy labeling we couldn't use, you say.
Yeah, so the heavy, so you couldn't in this case you can't heavy label the tumor, but you can heavy label your cell lines. And if you want to study breast cancer, you can heavy label many different cell lines and mix them together, and then mix that in with your tumor as a control. So you can have, let's say you have 100 tumors, and you have grown up, you have this mixture of cell lines that are all heavy. You can take a little bit and mix into each, so that each tumor, when you analyze them, so you get an internal control, and you can then use that to normalize all your samples. So that's so these are, I mean, different tricks to overcome this difficulty with quantifying. So now you can, people have tried to introduce heavy labels at every stage of this experiment. And probably one of the first ones came out of Rudy Ebersol's group called ICAT, which could be used to label proteins or after digestion peptides. But here you for example label all the cysteines in all the proteins, and then you mix them together after labeling. And now, so we're going to look at a few examples where we introduce the heavy label later and later. And of course, there are more and more. Anything before we mix them together, we have to be really careful that there is not a lot of variation. So you can do it on the protein level, and in this case, it's symmetrical, since in both cases we compare two samples that we label heavy and light. So we have a case where we shouldn't have too much systematic error because of the symmetry. Now we could make recombinant heavy proteins. Now we can't do a lot of these, but a few. But now we have an asymmetrical case, in the sense that in one case the cells go through lysis, while in the other case we just mix in the heavy proteins. So here, of course, we can get quite large systematic shifts, because of this lysis might be more or less successful in different samples. So we really have to then optimize how we're doing that. So then we have another where we use recombinant proteins that we label with heavy labels, and in this case these are not real proteins, but they are chimeric proteins. So we take, let's say, peptides from 20 different proteins that we want to study in our sample, and we make a protein that contains those peptides. So we make a chimeric protein with just a few peptides from each of the proteins that we want to study. So this method is very good for studying protein complexes when we want stoichiometry. Because here, by definition, we have the same amount of each peptide, since they are come together, and then we digest them together. The big question is, of course, will the digestion work exactly the same way in the proteins in our sample as in our chimeric protein. To make that more likely, what people do is they take the tryptic peptides and keep a few of the amino acids on each side, so that at least the local amino acid environment will be the same in the natural protein in the sample as in the chimeric protein. And then after mixing we can get stoichiometry. Now, after digestion, we can always also label. We can also label during digestion, with heavy and light, and then mix it together. And this is again a symmetrical case where we compare two different samples. But again, so now we have more and more of the steps before mixing that we have to be careful that they're not varying too much. Then we have another case where we make synthetic peptides that are heavy, and we mix them in after digestion. And then again, this is often combined with also with a peptide antibody enrichment to make the method more sensitive and to be able to look at more low abundance proteins. But again here, because of the asymmetry, that our sample goes through these steps, including the digestion that can sometimes vary quite a bit, and we mix in our heavy peptides. Because of this, we can get quite large systematic errors. So this has been proposed that it can be used to do absolute quantitation to really measure how much there is of the peptide. But because of these problems, it still can be quite a rough estimate of, and we can easily be off by an order of magnitude or two from the absolute quantity. But it's still very good for relative quantitation.
I feel like I'm not following because it's the point of having light and heavy. The difference is because we want to identify the control sample from the experimental sample.
Yes, sorry. So maybe I wasn't clear on that. So in the end, so in the mass spectrum, every peptide you will have one isotope distribution that corresponds to the light and one that corresponds to the heavy. And then we can use these relative intensities to determine what the relative amount is of the two corresponding proteins in the sample. And the nice thing is that we will have the measurement, of both in the same sample. In the same spectrum.
Yeah.
So that's.
So what's the advantage of this compared to if we measure the sample differently?
So you mean if we measure compared to label-free, for example?
Yeah, because we can, in that way, we don't need the light and heavy difference.
Yes. So the problem is that it's very difficult to keep the conditions constant. So by adding an internal standard, we have a much better chance to get a smaller experimental variation. That's really the main thing. And that's why people have tried all these different ways to do that. But it really comes down to that we want to try to minimize the experimental variation.
It's easier to quantify over time too, right?
Yes. And so for example, if you want to have a lot of samples, so it takes you months to measure them. By having some kind of internal control will make you much easier be able to normalize over a long time. So then now, so far we've only looked at, by measuring the precursor peaks, the peptide peaks, how to quantify. But now we're going to look at a few cases where we go a step further, where we then fragment the precursor and look at a few of the fragments. So the advantage here is that if you have a very complex sample, the full scan mass spectra where we measure the peptides will be very crowded, as we've seen before. So there will be a lot of high probability for interference. So now we're going to take a small window here and isolate this isotope distribution that might or might not have interference. And then we spread out the fragments over the whole mass range here. Then the probability of having interference will decrease. We'll still have, in some cases, interference that we'll see later in the MSMS, but it's a smaller probability. So by spreading it out, we make it cleaner. So this is the label-free case where we look in MS, and more commonly what we do is we add in synthetic heavy peptides to each sample, and then look at, so this is in MS, we see the ratio, but we then fragment each of them and look at a few of the fragment ions. And this is quite a common strategy nowadays to look at, to quantify a targeted set of proteins. Now, an additional way to look to quantify an MSMS is to what's called using isobaric peptides. So here we label after digestion, either with heavy or light, or with, usually in this case we can have multiplex several. So there are kits do up to 10. But these labels are isobaric. So when we measure the precursor mass, they will all have the same mass totally. But then they will have a weak link built into them, and a different mass will fall off. So what we get is reporter ions, usually at low mass, that correspond to the different samples. And since they are low mass, there's usually very little interference, and they can be very close to each other. So the first one that came out was a 4-plex called iTrack, which had the reporter ions were at mass 114, 115, 116, and 117. And then there were several other that came in, and now one can do a 10-plex, so 10 different samples together, and then with these low mass reporter ions. So the nice thing with this isobaric is that the full scans, they don't become more complex, even if you mix many samples together, since their labels are isobaric. And then the reporter ions show up in a part of the low mass end of the mass spectrum where there aren't many other ions that would interfere with them. And also they have been done in a way that at the same time, so if we look at the low mass end, we get the quantitation, and then the higher part of the mass spectrum will have the regular fragments that we can use to identify the peptides. So here now we have the information that we use for identification and the quantitation information in the same spectrum, which is very convenient. Okay, so we've gone through a lot of different ways to do this quantitation, and the one can use these different techniques for different purposes, but there are a few that are more common than others. But in general, when we then analyze the data, what it comes down to is to find either isotope distribution or find the reporter ions. So we talked a little bit about this already before. So one thing for a lot of these, especially for the MS1 quantitation techniques, we need to know about the isotope distributions. So again, when we have at lower masses, the carbon-12 peak is the dominant, and then when we go up in mass, there are higher isotopes that are more dominant. And we can calculate these, how the relationship between the ratios between the first two peaks and the first and the third peak is, and so on, and for the ratios between all of the peaks. So now, to estimate, we've talked about this also a little bit before, how to estimate peptide quantity from looking at these peaks. And the first thing is to find some kind of baseline, and then we can have options either to look at the peak height, either fit a curve, or the peak area. And most commonly it's either the peak height or the peak area that we look at. And then we have the time dimension that we should also use, so that we know that if we look at one mass spectrum, we see these peaks, but also it should be continuous in the time dimension, that first there is no peak, then the different peaks in the isotope distribution should appear at the same time and increase and then disappear together. We also talked about sampling, that we actually need surprisingly few points to get a reasonable estimate of the area. And this just shows that in the case where we only have 3 points on the on a peak, in about 85%, so in 95% of the cases, we get an estimate that's above 0.87 instead of 1. So even with such few 3 points, we can get a reasonable estimate. And then this just shows that when we come up to 4 points, we have probably most of the time more than 95% of the cases we get a good estimate, and so on. Now another issue we have to worry about is retention time. So if we run the same sample twice and then compare where different peptides eluted after how long time, we see that we can get, so this is the retention time, so this goes from 0 minutes to 120 minutes. And this is the retention time difference, goes from between -2 minutes to 2 minutes. So we see that there is a trend here, but sometimes we can get quite a large deviation. But we can fit some kind of smooth curve to this to get to subtract away this difference and get much better accuracy. This is in one case. This is another case where the deviation is even higher, so this goes up to 4 minutes. But again, by fitting a curve, we can subtract out the difference. Yeah, and this is something when we run many samples, we do want to do this to be able to compare, and especially for label-free quantitation, we have to make sure that we compare the same peak to the same peptide to the same peptide in both cases. So now there is one other way to estimate quantity, and that's by spectrum counting. So what we uh, what John Yates' group showed quite a few years ago is that if you look at copy number of a protein per cell against how many times we see a spectrum that corresponds to a peptide that is in this protein, there is definitely quite a correlation between these two. And the correlation becomes even better if we look at compare the spectral count for the same protein in different samples. Now the drawback of this is that it doesn't work very well if we have very few spectra. But if we, for proteins where we see a few peptides and a few spectra per peptide, it works reasonably well. So if we compare these different ways to estimate quantities, so the peak height is resistant to interference, but it has poor statistics because we only choose one point. Now peak area gives us much better statistics, but it's sensitive to interference if there is another peak that partially overlaps our peak. Now curve fitting would work well, but it's actually not used very much in practice, probably because it's quite slow. Now the spectral counting is very much resistant to interference because we require now that there is an MSMS spectrum that have enough fragments to allow us to confidently identify a peptide from that protein. And it's very easy to implement. That's probably why it has become very popular, because any output from any search engine, you can write an easy script to extract, just count the number of times peptides. There are spectra that correspond to a certain protein, so that's and that a lot of people can do, but for low abundance proteins, it has its limitation.
But with this spectrum counting, we still have to specify the error allowed, right?
Yes. Yeah.
So there is still a possibility there is something interference.
Yes, but it's much smaller. It's yeah. So definitely I think the smallest risk for an interference is in the spectrum counting method. Compared to the other methods, they're much more sensitive to interference. So then, just to look at a little bit more data. So this is one example from QTOF, a peptide isotope distribution. This is a function of time here. So first the peaks start appearing, then they grow, and then they disappear. And this is a very clean case when there is not much interference. Another case here, it starts out well, but then as they disappear, we see that there is another peak uh, coming up here, and finally we have a very different distribution here. So there is an overlap. So, and if we want to do quantitation, we have to make sure that we can separate these peaks out. Then we have another case where we have much more noise, and the peak intensities are smaller. So we see that we get much more variation. For example, you can look at the relative peak intensity of these first two peaks, which jumps back and forth just because we don't have very much statistics. And then another case where we really have a problem, and it's difficult to say what's exactly going on. There's definitely more than one peptide here, and then here we really need to decide which how to do the quantitation, or maybe this is a case where we just want to say that we can't use this for quantitation. Then if we go to a more high resolution instrument, we get a better separation between the peaks, and the chance for interference goes down. But also here in another area we see that this is our, where the three red lines is our peptide of interest, but there is actually also a lot going on here, and we don't really, the three peaks don't correlate very well with each other as in function of time. So even when we have very high resolution, we will get interference. So this is just another example of a peptide. So here we're looking at the first isotope peak, the second and the third. And so we see that we have, it's a nice case, the three peaks are well correlated. And if we look at the ratio between the first and the second and the first and the third, they're constant for the duration of the peak. And even after when it looks like the peak has disappeared, we get a pretty good ratio. But we're starting to get more variation there, of course. But as you see, there's very little. I mean, by eye at this linear range, we can't really see the peak anymore here. And also looking at the mass to charge ratio, we see that it's quite constant throughout the region. And this is what we can then use both these ratios and the mass accuracy to determine whether we have interference or not. So for example, here is another case where we have different, for this peptide, there is a peak here and then two other peaks out here. And we don't initially know what this is, and we see that for all the three isotopes we have these three peaks. And then when we look at the ratio, comparing to the ratio we expect for this peptide, we see that this peak actually has a ratio that's it's reasonably close to what we expect, while these others two peaks have ratios that are off. So these are the kinds of things. And also the other thing we see is that the mass error is slightly positive in this case for these three, and in this case for these two it's slightly negative. So, and actually what turns out for this whole LCMS run, the correct peaks were a few parts per million off in the positive direction, because the calibration hadn't been done better than that. So I mentioned interference before, and the main thing is that, especially if you look at low abundance proteins, the chance for interference is high. And interference meaning in MS1 it would be that there is a different peptide with a different sequence but with similar mass. And that at least partially overlaps with the isotope distribution of the peptide that we're interested in. And in MS, we can also get interference by it's a different sequence that has the same precursor mass and a few fragments overlapping. And especially if it's the fragments that we are interested in. So this is just showing different examples of, so in this case, so let's say this is our peptide of interest, the blue one, and we're looking at different peak heights, so the concentration goes down in this direction. And then we add on to this more and more interference. So what we can see then is we get more and more deviation from what we would expect, and higher error. So we look at this a little bit more in if we look at MSMS. So this was a data set taken from CPTAC, Verification Work Group 7 study. So they took 10 peptides and they measured 3 fragments, so 3 transitions per peptide at different wide concentration range, and they added it into human plasma background. And it actually was a lot of 8 different laboratories and 4 repeat analyses per lab. So we just took this data set and looked at it. So a lot of times we have a case like this. So this is the actual concentration and this is the measured concentration here. And all 3 transitions behave very nicely and fall on a line, a straight line, and we get a little bit more variation towards low concentrations in these 2 cases. But then we also have other peptides where we have 2 transitions behave nicely, but the third one deviates because of interference. And then we have a third case, which is that we have this deviation in one transition, and we also have that the systematic shift away from the straight line. And this is what I mentioned earlier, that you can get when you you digest your sample and you mix in your synthetic peptide. Then you can get these systematic errors because of the asymmetry of the experiment. So now if we take these 2 cases, the one that behaves nicely and the one that has an interference, and then now plot the ratio between transitions. So here we look at the ratio between transition 2 and transition 1, and the ratio between transition 3 and 1. So we see that over the whole concentration range they are constant. And when we do the same for peptide 3, we see that the ratio between transition 3 and 2 are constant, but transition 1 and transition 2 we get this variation over the range. So this we can now use to try to detect what's an interference. So but we did a simple model with noise and interference. So we have noise where the peaks move up and down between different measurements, and then we also have an interference which is always positive. So the noise goes both ways, and the interference is always positive. So now if we what we can do is then decide on how we can threshold this. So what we can do is to calculate the z-score. So and decide, and then we have to choose what threshold to use. So these z-scores were calculated by taking the maximum of the z's calculated for each pair of transitions. That is the deviation between the ratio that we expect and the actual ratio between intensities, and divide by the standard deviation. So now the the expected ratio we can do, since we have synthesized our peptide, we can measure these in for the relative. We can measure the expected ratio by measuring the peptide without a matrix that has a lot of interferences, and the the question is then how to calculate the standard deviation of the or the variation of the um, intensities, and that we get from our repeat measurements. We can calculate that, and the other thing is that we always use log intensities for these calculations. So now a little bit about the simulations here. So this case we have now noise but no interference. So we're gonna so what we get is the corrected relative error. There will be the distribution will be widened because of the presence of the noise. So now we can set different thresholds for the z-score. And so this is now 3 transitions, just relative intensity is 1 to 1 to 1, and the relative noise is 0.2, no interference. So what we're going to do is, if we change and set a very low z-score threshold, we're going to get a distortion of this distribution, and it's not very good because our estimate will be deviate from what it should be around, symmetrical around 0. And so if we start with that case, we can plot against the threshold, we can plot what the centroid is, and then as we move up in thresholds, we can see that in this case somewhere around 2 we get a reasonable centroid. We still have a tail here, but it still gives us a pretty good estimate of in the case. So now again, remember, we haven't introduced interference yet. So now we're gonna look at interference, but first, and we're going to look at it, plot it in here, where we on the x-axis have relative error, and then before correction, and then we have relative error after correction. So so if we have no correction, then we're going to have the relative error before and after correction are going to be the same. So there's no change. So then we would get the line here. And then if we do a perfect correction, whatever our relative error is, the corrected relative error will always be 0. So we will get, so this is of course what we strive for, but it's, as you see, we're not going to reach that perfectly, but we're going to try to get as close as possible. So now here, now we're going to have both noise and interference. So now, first, if we don't do correction, we have these points along the diagonal as expected. And so again here we have the same relative intensities of transition 1 to 1 to 1, relative noise 0.2, and one out of 3 transitions there is interference in. So now we do correction. So for some reason, I guess these pictures are a little bit big, so there's a delay. Okay, so now we go from this case where we have no correction. We come down, if we do a correction with the threshold of 2, then we see that we move down all the ones that have large relative error. We move them down to smaller, we do correct all the ones that are up here before correction. They move down here. So we do reasonably well with correction. So here, these smaller ones, there's no correction for the ones that stay in but they are relatively small anyway. So now this is the same case. If we have very low threshold, we do correct, but we overcorrect. So we get, you see that this moves down. So we get an asymmetrical case. But then as we move up, you see that around 2 we get a pretty reasonable correction. We see that we get, again, it's a compromise between, because we get a little bit longer tail still down here than we would want, and we get this small bump of the ones that are not corrected, that are out here. But all in all, it seems like a threshold of 2 is quite a good compromise. And this is the case if we go higher. Now, if it's at the threshold to 3, we get a big bump here. And so this was in this area around here between 0.3 to 0.7. And then if we look out here in this area, 1.3 to 1.7, we get quite similar, that the threshold of 2 works reasonably well. So now this was the previous case where we had interference in 1 out of 3 transitions. Now, if we have interference in 2 out of 3, we can still do the correction, but as you see here, we get a much bigger spread. So we introduce much more uncertainty. So now this is a threshold of 2. So if there are 0 interferences, then we introduce this tail by setting the threshold. So we make it slightly asymmetrical. If we have 1 interference, we make it becomes slightly wider distribution, and then if you have 2 interferences, it becomes a lot wider. And that's the same as we see here. So that's just looking in this gray area here. So now that we've looked at, these were so far just simulation. Now we can go back and apply this to real data. And what we can do is we take this case where 2 transitions behave well and the third one not so well. And if we just take the average intensity, the blue is the uncorrected. So we see that we get this deviation. But then the red triangles are the corrected, and we then get a much better measurement down to low levels. And then this is just looking at the different ratios, and we see that there is this interference in I guess in 2, in transition 2. Well, so this is the heavy labeled standard, which doesn't show a. So to summarize, quantitation with LCMS, so what we need to do is to first standardize the measurements, as we mentioned before. So we need to align the retention time. Potentially we need to recalibrate the masses, and we need to do intensity normalization. And that we're going to talk about later when we go through more in detail how we would analyze the data from these kinds of experiments. Then another thing we need to do is quality control, because what often we need to recognize cases where something really went wrong. So we need to have some kind of way to see that, because those data we have to then throw away. So there are all sorts of things that can go wrong. Probably the most common one is that the chromatography column clogs and we don't get anything out. Or that the spray sputters and there is not a good spray. So these are things that we should implement different ways to recognize. Then when it comes to quantitation, we talked about that how we need to be able to find the peaks, how we subtract the background, how to set the limits of integration for time and mass, and how to exclude interfering peaks. And this is sort of the summary of what we need to do good quantitation. And so to just summarize in a different way, there are many different ways to do quantitation, and some are more appropriate for some applications than others. And it's also often a question of cost that has to be, and time, how long time it will take to, because one can always design an experiment to that it will take a very long time to analyze each sample, but then if one has many samples, one has to make a trade-off so that it's doable within the time that's and in general uh, to make the experimental variation as small as possible between different analyses, it's best to introduce the isotope label as early as possible in the experiment. And then it's important to always monitor for interference, because we're usually looking at very complex samples with many proteins that we then digest to make many more peptides, and often we're interested in very low abundant proteins there also. So that's the end of today's lecture, and any